Imagine a bustling city, alive with activity. The streets are filled with cars, buses and people, all moving with purpose and direction. Buildings rise, roads are paved, and energy flows through the veins of the city. This constant motion and construction are essential for the city's growth and maintenance. This is what the inside of our cells is like, a microscopic world where proteins are the workers that make life possible. Just like a city, our cells are bustling with activity, ensuring everything runs smoothly. Proteins are the building blocks of our bodies, responsible for everything from fighting infections to digesting food. They play a crucial role in almost every function our bodies perform. But where do these essential proteins come from? How are they created within our cells? The answer lies in a fascinating process called protein synthesis, a fundamental process occurring within every living cell. This process is vital for the creation of new proteins. It's a complex symphony of molecular machinery working together to create the proteins our bodies need to function. Each component has a specific role, contributing to the overall harmony. This intricate dance of molecules ensures that our cells have a constant supply of the proteins they need to thrive. Without this continuous production, our cells would not be able to sustain life. At the heart of protein synthesis are ribosomes, tiny organelles found in all living cells. These microscopic structures are essential for life, playing a crucial role in the production of proteins, which are vital for countless cellular functions. These remarkable molecular machines are the protein factories of the cell, responsible for assembling amino acids into long chains that fold into specific three-dimensional structures, forming functional proteins. Without ribosomes, cells would be unable to produce the proteins necessary for growth, repair, and maintenance. Ribosomes are made up of two subunits, a large subunit and a small subunit, each composed of ribosomal RNA, or rRNA, and proteins. These subunits are intricately designed to work in harmony, ensuring the accurate translation of genetic information. These subunits work together like two hands, clamping onto a strand of messenger RNA, or mRNA, and using its instructions to assemble proteins. The process is highly coordinated, with each subunit playing a specific role in decoding the genetic message. The small ribosomal subunit binds to the mRNA molecule, while the large subunit joins amino acids to form a polypeptide chain. This chain will eventually fold into a functional protein, ready to perform its role in the cell. Think of the small subunit as the reader and the large subunit as the builder. Together, they ensure that the genetic code is accurately translated into the proteins that sustain life. The instructions for building proteins are stored in our DNA, located within the nucleus of every cell. However, DNA never leaves the nucleus. So, how does this information reach the ribosomes in the cytoplasm where proteins are made? This is where messenger RNA, or mRNA, comes in. mRNA acts as a courier, carrying genetic information from the DNA in the nucleus to the ribosomes in the cytoplasm. The process begins with transcription, where an enzyme called RNA polymerase reads the DNA sequence and creates a complementary mRNA molecule. This mRNA molecule then travels out of the nucleus and into the cytoplasm, carrying the genetic blueprint for a specific protein. Once the mRNA molecule reaches the ribosome, the process of translation begins. Translation is the process of decoding the mRNA sequence and using it to assemble a chain of amino acids. The mRNA molecule carries the genetic code in the form of codons. 3 nucleotide sequences that specify which amino acid should be added to the growing polypeptide chain. The ribosome reads the mRNA codons one by one, matching each codon with a specific transfer RNA or tRNA molecule. Each tRNA molecule carries a corresponding anticodon, a 3 nucleotide sequence that is complementary to a specific mRNA codon. Transfer RNA, or tRNA, plays a crucial role in protein synthesis by acting as a shuttle service, delivering the correct amino acids to the ribosome based on the mRNA codons. This process is essential for translating the genetic code into functional proteins, which are vital for countless cellular functions. Each tRNA molecule has 2 important regions: an anticodon that binds to a specific mRNA codon, and an amino acid attachment site that carries the corresponding amino acid. The anticodon ensures that the tRNA matches the mRNA sequence accurately, while the attachment site ensures the correct amino acid is delivered. When the ribosome reads a codon on the mRNA, the tRNA with the matching anticodon binds to the ribosome, bringing its attached amino acid into close proximity with the growing polypeptide chain. This precise interaction ensures that the amino acids are added in the correct order, forming a functional protein. Think of tRNA as a delivery truck, carrying a specific amino acid cargo to the construction site of the ribosome. Just like a truck delivers materials to build a house, tRNA delivers the building blocks needed to construct proteins, which are essential for life. The process of protein synthesis is like building a complex structure with Lego bricks, where each amino acid is a Lego brick, and the mRNA codons are the instructions. The ribosome moves along the mRNA molecule 1 codon at a time, and for each codon, a tRNA molecule with the matching anticodon delivers its amino acid cargo. The ribosome then catalyzes the formation of a peptide bond between the newly delivered amino acid and the growing polypeptide chain. This process continues, codon by codon, amino acid by amino acid, until the ribosome reaches a stop codon on the mRNA, signaling the end of the protein. Once the polypeptide chain is complete, it detaches from the ribosome and undergoes a process called protein folding. Proteins are not functional in their linear form. They must fold into specific 3-dimensional shapes to carry out their biological roles. The sequence of amino acids in a polypeptide chain determines how it will fold, as different amino acids have different chemical properties that influence their interactions with each other and with the surrounding environment. The folded protein then assumes its active conformation, ready to perform its specific function in the cell. Protein synthesis is essential for all aspects of life, from the smallest bacteria to the largest animals. Proteins play countless roles in our bodies, acting as enzymes that catalyze metabolic reactions, hormones that regulate physiological processes, antibodies that fight off infections, and structural components that provide shape and support to our cells and tissues. Without protein synthesis, our cells would not be able to function. Our bodies would not be able to grow and repair themselves, and life as we know it would not be possible. While protein synthesis is a remarkably accurate process, errors can occur, leading to the production of misfolded or non-functional proteins. These errors can be caused by mutations in the DNA sequence, errors during transcription or translation, or problems with protein folding. The consequences of errors in protein synthesis can range from mild to severe, depending on the protein affected and the extent of the error. Some errors may have no noticeable effect, while others can lead to genetic disorders, developmental defects, or diseases such as cancer. Protein synthesis is a fundamental process that underscores the complexity and elegance of life at the molecular level. From the intricate machinery of ribosomes to the precise delivery of amino acids by tRNA, every step in this process is a testament to the power of evolution to create sophisticated molecular systems. Understanding protein synthesis is crucial for advancing our knowledge of human health and disease. By unraveling the intricacies of this process, we can develop new strategies to combat diseases caused by protein defects, engineer proteins with novel functions, and gain a deeper appreciation for the molecular symphony that sustains life.